Hello, everyone. Welcome to the Careers in STEM series. We are FRC Team 226 Hammerheads from Troy, Michigan, consisting of over 100 high school students from Troy and Athens High School, and we've been competing in the first robotics competition since 1999.
This is a bi-weekly show occurring on the first and third Thursday of every month. For the series, we'll have a 30-minute interview period and then a short Q&A at the end for audience questions.
My name is Shaun Dantas, and I am a junior at Athens High School.
My name is Sonnet, and I'm a sophomore at Troy High School.
Our guest today is Meha Patel. Meha graduated from Troy High in 2015 and majored in cellular and molecular biology at the University of Michigan. She's interned at several companies and is currently pursuing a post-baccalaureate degree at the NIH. So, Meha, could you tell us a little bit about what you did in high school?
Hi, yeah. First off, thank you for having me. And I guess in high school, I kind of bounced around from different topics. I remember freshman year, I thought I should try a little bit of everything, mainly having that mentality that if you don't figure out what you're doing in high school, you won't be like ready for the rest of your life, which is completely false. So I think in high school, I did a lot of APs. I started a couple of my own clubs at Troy High. I don't know if they're still there today. And I guess extracurricular wise, I did student government, Model United Nations, robotics, which was one of the best highlights. I also participated in language clubs. So I was in Chinese club for a little while and then French club. I did do a quiz bowl for a couple of years. And then in addition to like the arts, which like were orchestra, choir, participated in sports as well. So I did cross country, and in my senior year, I was also on the dance team. But yeah, I did in the environmental club, which I think their name changed, but bounced around a lot.
Yeah, it seems like you had a lot of really cool experiences across a lot of different clubs. But could you elaborate and tell us a little bit more about your time on 226?
For sure. So on 226, I actually joined as I want to say it's a fluke. Srini, who kind of recruited me, just said, hey, you should try this out. I think it was like my junior year. And basically, I started off on the robotics side, which was interesting. I always had it in my mind that I should follow engineering like the rest of my family. So she was like, why don't you try it? And I think I was putting together a chain or something, and I was like, this is not for me. So within 2 weeks, I think Srini and I forget who he was working with at that time. I think Jeff decided that we should have a business side of the organization, and I quickly transitioned from that to the business side. So it was a lot of like getting to know people, talking to other teams, cheering on our team. So I did a lot of that for the organization. I worked with the chairman's group a lot, and anything that had to do with spirit, that was our job.
What was like your most memorable moment on the team?
Oh, I think it was the Troy meet our senior year. I think we were setting up at Athens, and we were putting up the welcome FRC teams on the fence in red, white, and blue plastic cups. It was cold. It was freezing. It was like starting to drizzle. And I think we ended up winning an award for that, which was crazy. And it was a memorable experience with a few people.
As a girl on the team or a girl in STEM in general, were there any particular challenges or difficulties that you had to overcome?
I would say, in addition to just trying to prove yourself, which is just the general, like, just of most females in any male-dominated field, especially being on the cheer side, you didn't get like taken seriously spirit-wise or anything. So really making a name for yourself and trying to like, I think one notable experience for me was trying to control the entire room in order to teach them some dances so that we wouldn't show up to a meet and not be able to participate in some of those displays. But getting everybody to take that kind of thing seriously, which I know it's not something you'd think about at an FRC competition, but trying to communicate was one of the hardest things. And then my partner at the time was Richard. I don't know if you've interviewed him yet, but he and I often butted heads, which was just like a part of trying to understand people, which you just have to work through. There wasn't really a handbook for me to follow, but I think we worked through it, and I definitely recall 226 as one of the most memorable experiences of my high school career.
So obviously, it seems like you had a great time with 226, but what was your major like moment of realization for picking a major? Because a lot of kids go through engineering and you didn't, you're an outlier, I'd say. So it was quite interesting.
Yeah, I do remember the moment I picked my major. I applied to the University of Michigan, and I got in, I accepted, and I went, and I believe it was like my, I think it was our orientation, and they were going around in a circle, and I was originally actually supposed to do bioengineering. I was going to move from the LSA school to which I had just gotten accepted to into the engineering after a year. And going around in a circle, I was just hearing everybody like talk about their majors. And I think it was like in a split-second decision, the person on my right just went and said their major. And I was like, you know what? No. I decided that I was going to do cellular, molecular, and developmental biology. So I just said that, and I was like, all right, sticking with it.
What does like cellular and molecular biology really entail? And besides like inspiration, like in the moment, were there any other factors that led you to pick that major specifically?
So cellular, molecular, and I actually tacked on the developmental biology my senior year. It's basically, in a nutshell, research biology. So instead of getting just to a basic understanding of all biology, you go into the cellular processes, you go into enzyme substrate interactions, chemical interactions, as well as more on the side of like problem solving. So you often, in most of my classes, would get case studies. So we'd be asked a question and you'd be given a toolbox. That's the main thing. You're given a toolbox and you're like, hey, figure out what the issue is in this case. So I'd say it's a lot of experimental, kind of mad science, which I love, which is why I actually stuck with it. A couple of my professors, as well as a couple of the like summer opportunities that I went through, just showed me that science was a cool and kind of unknown field that we really didn't know. And I think there were a lot of factors that led me to commit to it, and like I fell in love with it completely, which was, I think most of them were like research based. So actually getting into a wet lab and being able to conduct research and see cells outside of the body, stuff like that.
So were you involved in like any major organizations during like at your time in the University of Michigan?
Yeah, so I was actually in Biological Students Association, as well as an MCGB small group, which mentored, I believe it's called MPAC now, so Michigan, I think it's like People Connected, I forget, but it's basically just like mentoring other students who are in probably their further year, in their undergrad years, so their lower level years, and basically telling them what opportunities were good for them and helping them find what your niche, because MCGB is like a wild range of topics. So my mentor actually, she's the reason I'm at the NIH, because she went to the NIH and she's currently here. She's actually at the National Cancer Institute. So she kind of like helped me out in that sense and got me ready for future goals.
Besides all the student organizations you were able to involve yourself in, were there any internships that you took part in while you were in school, and how did you go about obtaining those opportunities?
So internships, I was I don't necessarily call them internships. I call them opportunities just because they're not as much as on the business side or engineering side as I'd like. I did a couple of like lab rotations at Oakland and then also at Michigan before joining a full-time lab. So at Oakland University, I worked with a professor on it's actually a physics-based project related to biology and medicine that dealt with cardiac rhythms. And then I think my most favorite experience is my undergrad lab, which was with the Batida lab, where we studied prostate cancer cells as well as proliferation quiescent decision, which is that entry into rapidly proliferating cells, which you see in cancer, versus quiescence, which is that metastatic cell type. So that's the silent killer kind of thing.
So, what is your what is your current position at NIH entail, basically?
So my current position at the NIH, I'm a fellow, which is a scientist that conducts her own research project, an independent research project. And what I do on a day-to-day basis is anything a regular scientist does. So I do experiments, I troubleshoot, I ask questions, I bounce questions off of other postdocs and doctors. And I like to think that I perform mad science because nobody's ever done what my project is before, which is completing an entire metabolic profile of B cells. So there's really a lot of like researching that goes into it, reading papers, contacting professors who are doing something similar, which you can hope and pray that it'll be applicable to your project. And then I also attend like meetings about papers and literature that are notable in science. So we talk about progress and all of our projects here at the NIH.
Since there's a lot of learning and problem solving that goes into your work every day, what do you think is your favorite and most fulfilling part of your job?
Ooh, I would say my most fulfilling part of my job would be the people that I meet along the way. I'd say that doing an experiment is great and having it work out is great, but getting to collaborate with some of the brightest minds that this nation, as well as like the world, has seen is amazing. You don't get this kind of experience, I'd say, every day. So like talking with some of the people here, meeting some of my I want to say like some of my I want to say like models and stuff like that. Just like in person. Like I think the first time I saw like Dr. Pierce, who was my boss, I was like, I've read all of your papers and I can't believe that I'm actually here with you.
So you just talked about your boss. Have you ever had some sort of conversation, or have you had like any relationship with Dr. Fauci?
My 1 second claim to fame with Dr. Fauci, which like after this pandemic started, everyone was like, "Have you met him?" I actually attended a conference, which was a small conference / talk, where a scientist was defending their paper at the main campus, and he was in the room. He asked a question, but I think it was we were less than like I want to say it was just like a couple of seconds where I was like, oh my gosh, that's him. And it didn't realize or occur to me that was actually him until after the pandemic started. And I was like, oh yeah, I've seen this guy before.
So as a post-bac, obviously you talked about being a fellow and what your day-to-day sort of looks like, but how is that sort of different than being like a postdoc researcher?
So the way that my project actually works is that I'm working completely independently from my mentor. It is a trial a trial in a sense. So I would argue that I am not towards the level of the postdoc. I work with them. Postdocs just have more training. They have more expertise in their field. I've only had about 2 years of expertise in my field. So the main difference would be that they're just well versed in what they do. And I'm just here being, I want to say that I'm like a baby of the lab. I'm just here being like, I'm going to try this out. And if I don't really like this topic, then I'll go and find my own topic. So they like, anytime I'm like, I don't know what's going on in this like one experiment, they'll be like, oh yeah, let me take a look at it. And like within 20 minutes, they're like, oh, this is what's going on. However, if I had tried that myself, it would probably take me like about 3 months or 2 weeks.
So, you said that you work at you're at the NIH right now. What happened on January 6th during the Capitol insurrection? Like, what was that like for you?
So the NIH is actually located in Maryland. So we were decently far away from I guess it's the insurrection that they're calling it. We were decently far away. However, we did all get the alert that the Capitol building had been stormed and that there was an issue and question of safety. So I was on the main campus at the time, and we went into lockdown, and we just wrote it out. We weren't sure what. Oh, sorry.
Yeah. With the pandemic and everything going on, has your lab made any like specific modifications? Are you guys still doing it in person? How are you guys transitioning?
So at the beginning of the pandemic, we went and decreased our numbers in lab physically, and tried adhering to all of the government guidelines. We tried I guess the staggered starting, which was one person goes in, we wait an hour after they leave. That one person is required to like sanitize everything they touched, and wear masks, gloves, everything. And that slowly progressed from like introducing one person at a time to 6 people in lab at a time, with masks, social distancing. Like you try and minimize as much contact with each other as possible. So a lot of us were like doing night shifts. I think at one point I was going in at like 11 p.m. and coming out at like 6 a.m. Just because like my experiments take a lot of time. And that's the only time that nobody's in lab.
Have you had any interaction with like the COVID-19 strains itself, or is that sort of independent of what you're doing?
So that would actually be independent of what I'm doing. My lab focuses on, it is within the immunology field, but the umbrella of my lab encompasses immunogenetics and activation. So what we do is more on the side of what is going on with your B cells, how are they being activated, how are they responding to different infectious diseases. Unfortunately, COVID-19 is not one of the disease models that I study. What I do study is malaria, LCMV, which is Armstrong infection, and I recently just got into the EBV field, so Epstein-Barr. So slowly branching out, but not COVID-19 yet.
Since as part of the immunology, you get to work with so many real-world diseases that impact so many people, is that part of the reason you like doing what you're doing? Like, how does getting to interact with those real-world issues make your job better?
I personally think that it makes it, you see a little bit more of a result. So in my old lab, I would work with this very rote scientific question: what is going on with prostate cancer in their decision to proliferate or enter that quiescent stage? Unfortunately, you don't see your research progress more than just that scientific question. However, with immunology, and what made me fall in love with immunology is that connection to real-time people who are undergoing these infections. So I worked on a project for a little while that is actually in a phase 1 trial for a cerebral malaria drug. So you see and maybe it's moving fast, but you see the transition to, you've studied something in a petri dish, and then now you're seeing it being applicable to patients who are undergoing these diseases. And it's really life-changing.
So what does like a regular workday look like for you?
Okay. Regular workday can range, but currently I go in at around 5 a.m. I'm conducting a survival study on mice. So we're looking at how long different strains of mice respond to malaria infection. So at 5 a.m., I go in and I basically I do a clinical scoring, which is looking at the mice, seeing how their motor, neuro, and in general, how are they looking? So if they're sick, if they're able to grasp, if they're able to walk, if they can, there's a little like muscular test that we do as well. So just doing all of those tests, and then I collect blood from them, which lets me measure their hemoglobin levels as well as their parasitic levels. So after that, I take all that data, go back to my lab, and I look at the mouse's blood cells underneath the microscope in order to determine how far along the malaria parasite has infiltrated their system, as well as compare the long-term data. So if they're getting better, if they're getting worse, and that's just one experiment. The other experiments that I do is I am comparing diets on infection models as well. So does a diet, like a Western diet versus a low-fat diet, affect how your B lymphocytes react? Which is super important to like vaccine studies. So I'm also reading a lot into that because I'm super new on that project. But mostly it's either in vitro work or in vivo work. So in vivo work is what I do in the morning with the mice, and in vitro work is what I do outside, which is like look at those blood cells underneath the microscope. I'm also trained in like doing microscopy. So some of the time I'm running tests on B cells that have been in culture and staining, sometimes looking at fluorescence. So what your B cells are doing in live time.
Since you have like something you do in the morning and something you sort of do in the evening, for your research time that you spend at the NIH, is it sort of split between two projects? And like what exact part of malaria are you studying?
Sorry, would you mind clarifying your question? Are you asking about the so like what portion of malaria am I studying, or like the long-term effects of malaria?
So like what portion of malaria you're studying?
Okay, so I would say I'm understanding the response of the host B lymphocytes and the disease progression in terms of that. So looking at immunity, how does it build? What is going on in terms of development? So a lot of the time, I will look at your at mouse spleen cells, peritoneal cells, as well as bone marrow cells. So going from like what are B cells doing from their infancy stages, as I like to call it, to their full out mature stages, and then also how they're responding under infection. So different types of malaria strains actually respond differently.
So, do you ever plan on moving towards maybe like a larger like pharma company where you plan on developing like your own vaccine after all your research on specific like disease or whatever you're studying at this moment?
I would say, although I love the infectious diseases that I have been able to study, I think that I'd like to transition to HIV/AIDS research if I did choose to go into industry. Mainly because I think HIV and AIDS just function so differently from any of the other viruses out there, as well as just that whole like you impact a lot of people who are not only socially stigmatized, and coming from a pre-med background, like that side's always interested me, being able to help people. But not only socially stigmatized, but also like life expectancy. They've been cut nearly in half and can it's just interesting, that entire virus. I don't know too much about it, which is also part of the reason that I'm just, I guess, curious by it and ensnared by it. So I would try and move to HIV/AIDS if that was my goal in industry.
When you were right out of college, how did you decide that you sort of wanted to first do like more of a research opportunity at NIH instead of working at one of those big companies?
So my senior year, I was actually still pre-med. So I had applied to medical school, and unfortunately, I was rejected in that application cycle. And it caused me to reevaluate, I think. So I knew I wanted to help people, and I knew I definitely, I needed to do something that was along that track in order to feel fulfillment. But my mentor at the time in my lab said, I think I know which way you're going. And she said, you can save a lot of lives by being a doctor. Sorry, you can save one life by being a doctor, but you can save more lives by being a scientist. Just thinking about like how many vaccines there are, as well as like studying just random things that happen with our body, like understanding just helps a lot of people. So she's like, she pushed me in the direction of the NIH and said, you might want to try something out here and see if you like a field, if you don't like a field. Because I was also very, I don't want to say directionless, because I liked a lot of topics back then. I just didn't know which one to choose. So she said, go and explore a little before you settle on a topic. And I think that was the best advice anybody's ever given me. Because immunology is just something I fell in love with by just being here.
So it seems like your mentors really helped you a lot, and I know a lot of high school students really, or even in college, I'd say they struggle with picking the major. Like a lot of people have directions they want to go to. So how did you go around finding your mentor or somebody that you could ask for help?
So actually in high school, I know myself, I did not ask for help a lot in high school, which I think I turned around in college. So in high school, I think a couple of my mentors were like Shriti, my biology teachers were amazing. I don't know if Miss Brewer is still there, but Miss Brewer was amazing in helping me determine that biology was one of my passions. I don't know if she still does the starfish things, but that was just amazing. I think just asking for help is one step. And I like in high school, I was shy. And I definitely I didn't like doing that, like reaching out and stuff like that. But once you do reach out to mentors such as Miss Brewer, my lab mates, my PIs, they open a lot of doors for you. And they also can help, like, because they've been through what you're going through. They've been through the curiosity phase, the determining where you're trying to like go, what questions you're trying to answer, and they're really helpful in finding a fit. So if I could give any advice, I would say do step over that, even if it's like scary to do so. It doesn't hurt to ask, and I know you hear that a lot as high schoolers. Like, there's no such thing as a silly question. There really isn't. You can ask any question and someone will be like, hey, yeah, I got you.
Since you came so far from high school and then all the way to where you're currently working, besides the suggestions that you just gave, do you have any other final advice that you have for our audience or anyone who wants to pursue interests similar to you?
I would say try everything. Try a little of everything. If you don't like it, you'll figure it out soon enough. So I think I went from student government to Model United Nations to Quiz Bowl to like to robotics. Essentially, I ended my high school career with robotics, which I think was amazing. And robotics encompasses a lot. So I would actually recommend people join robotics a lot earlier than I did, just because 2 years was not enough with the team. But just trying a little bit of everything. So that might be doing orchestra, that might be taking that extra AP class, that might be dedicating an extra hour to whatever sport. For me, that was like dance and then cross country and then karate, but going that extra step, which can be scary. I know it's kind of crazy to go against the status quo, but just doing a little bit of everything.
Well, thank you so much, Meha. we're now going to be moving on to a few questions from our audience.
Yeah, so one of the audience members asked, what is it like to work in Washington, D.C.?
I would say it's an interesting take, especially being in the nation's capital. You get exposure to a lot of politics, and working for the government is interesting in the sense that not everybody might share the same political views as you, but you still have to interact with people. So it teaches you a lot about people skills. And I wouldn't say we skirt around politics. We're very vocal, but it helps you not only you're not only doing science, you're also learning a lot about politics, a lot about people from all walks of life. So I want to say that Washington, D.C. is a really vibrant city, where you can essentially get into anything. There's a lot of us who tried different things for the first time. So one of my coworkers is trying horse riding. One of them does scuba diving. It's really crazy. There's a walk of life and a group of people for anybody here. And I wouldn't consider it a huge city like New York or anything, but it's interesting. You just get a lot of backgrounds, and a lot of people who you might not necessarily like at the first view, but who you get to see past a lot of different perspectives.
Do you think working at Washington, D.C. and interacting with all these people from a lot of diverse backgrounds has given you a more unique perspective?
Definitely. I want to say when I came here, I was just very focused on doing science. I was really ready to commit to this question that I wanted to answer and just that question. But looking back, I've branched out in so many different directions, that I don't have just one project. I've been working on several, and that's just in at work. But also outside of lab, I've met and made friends with people who I never thought I'd meet. So just I think it's definitely broadened my horizons in that I can say that I look at a situation from different views, than just the one that I was immediately privy to, which does help in a lot of cases, because sometimes it's literally putting yourself in others' shoes, but that only gives you two views, yourself and that person, but then seeing it from a bystander point of view, which is how I'd like to characterize it.
Outside of work, are there any particular projects that you're working on that are really interesting?
Outside of work, I am currently, so I was working with a group of, previously I was working with a group before the pandemic, that was helping distribute medical kits to DC's homeless, as well as testing kits to the LGBTQ populations in DC, just because of stigma associated with going into a medical provider and not getting service. That was a project that I was working on before the pandemic. However, when the pandemic started, a group of post-Baccs and I got together, and were basically trying to solve a lot of the logistical issues that were occurring in our communities. So I, along with another post-Bacc, founded a COVID-19 response team, which was aimed at assisting populations that got hit by COVID, but didn't necessarily get the same, resources that everybody else got. So this dealt with a lot of the homeless, sometimes domestic abuse, and people like that. I'm currently also trying to become an EMT, so we'll see where that path takes me. But those are my projects outside of lab.
Since the pandemic has been going on for a couple of months, how has that your work with COVID-19 sort of shifted?
So in regards to the group that I've helped start, we were basically, before the pandemic and all of its strict volunteer guidelines that got in place, we were assisting in distributing masks, helping homeless people get to safe locations that they could ride out cold weather with. Because a lot of soup kitchens, homeless shelters, and churches, as well as like centers for providing for domestic abuse and stuff, closed cold turkey. Nobody was able to do that. So we were looking for places to partner with in order to assist these populations in any of their needs. So that could range from getting food on the table to essentially helping someone get out of a situation. The last time I heard that project was transitioning into being incorporated into George Washington's DC Cares project, which is a website that is aimed at combining all these resources into a one place, one stop location for anybody to look up what they need.
Well, thank you so much, Meha, for talking with us today. It was really great seeing your insight, and I think you bring a really good outlook that we don't get enough on, or don't get enough of on the robotics team, because you really bring a new perspective on stuff. So thank you, everybody, for joining us today. Remember to subscribe to our YouTube channel, and come back on March 4th for our show with Meha Patel. He's an aerospace engineer at the Aerospace Corporation who graduated from Troy High in 2016. We hope to see you then, and thank you for watching.